Thank you very much for coming to hear the first Life Science Speaker Series talk. As Elaine was saying, my name is Brandon Guida. I always like to start off, especially in the student pool, the demographic that you are, and just kind of give some background on myself and how I got to where I am and some of the difficulties that I've had along the way. I originally am from New York. I grew up in Arizona, though. I moved here when I was a kid. I've done my high school was in Glendale. I kind of always knew I wanted to be in science, but I didn't exactly know what. And I think a lot of people fit into that. They find a subject in school and they're like, I really like this, but what the hell am I going to do with it, right? What am I going to be when I grow up, so to speak? And in certain aspects, I still don't know what I want to be when I grow up. I'm just kind of doing what I'm doing right now because I love what I do. So I went to NAU, Northern Arizona University. I got a bachelor's in microbiology. I thought I wanted to be a surgeon, a doctor. And so I started getting into a little bit more of the research aspects up there as an undergraduate researcher at NAU. Found out I really loved bench work. I really loved science. I wanted to actually do science. I wanted to see how things worked. Worked in infectious disease for a while. I worked on anthrax up there at NAU. And at the time, I was working on anthrax was 2000, 2001. What happened in the United States along that time period? 9/11, right, 2001. And what were people getting in the mail? Anthrax, right? And so there are really only 2 or 3 labs in the United States that work on anthrax and work on weapons-grade pathogenic anthrax, and our lab was one of those. So we got visited by the FBI, and we all got questioned, with the light, what were you doing on this day? Have you had any contact with terrorists? All that kind of stuff. So that was probably the highlight of my undergrad career. And I'm like 18 years old, and I'm like, what's going on? So that was crazy. And then after that, I didn't know what I wanted to be when I graduated. So I got a bachelor's in microbiology. And at many universities, when you get a bachelor's in science, oftentimes you get an automatic minor. And my strongest advice to you as a potential bachelor's student is make that minor a double major. It's 3 extra classes a lot of the time. So I had a chemistry minor, and I took the 3 extra classes, and I got a biochem dual major. It's wicked simple to do, and it's just a couple of extra classes, and it just looks that much better on your resume. So piece of advice. And then after that, I really didn't know what I wanted to be. So I said, okay, well, I'm going to go work for some pharmaceutical companies and see if I like the gig, if I like being in the biopharmaceutical industry. I worked for Pfizer, worked for a local company called Watson Pharmaceuticals. I worked for Roche, which is a really large biotechnology company. Didn't really care for it. So I wanted to go back into academia. So then I went, found a master's program where I could get 2 masters in 3 years. I did that back east at the University of New Haven, and I got a master's in molecular biology and a master's in bioinformatics. That's what this MCS is, if you're wondering. It's a master's certification in science. It's a non-thesis master's degree. It's basically coursework. Right. So I got a master's in molecular and cellular biology and a non-thesis master's in bioinformatics, which is like computational biology using computers to analyze complex biological data. And then after that, I said, I love science. I want a PhD. And so then I came here to ASU, where I began my dissertation research in infectious disease. Once again, I was working with Mycobacterium tuberculosis at the Biodesign Institute at ASU. And then things didn't work out there, and I switched labs, completely switched fields, ended up in environmental microbiology somehow. And I worked with some bacteria that live in the ocean. Totally unrelated to things I wanted to, but that's the way science works. Sometimes you fall into a gig and you just go with it. And so after that, I said, well, I kind of want to go back to neurobiology, which was kind of where the focus of my molecular and cellular biology master's was. And that's where I ended up, and that's where I am now. So I work in Dr. KRUER's lab. He is a physician scientist. We are a translational clinical laboratory. So most of the time when you work in the sciences, you're doing bench work, you're publishing papers, but usually that science that you're doing takes a really long time to come into practice, right? It takes 20 years and $2 billion for a drug to hit the market. That's why drugs are so expensive in the pharmaceutical industry. So a lot of times it takes a long time for the science that you do as an academic scientist to make any kind of benefit to humanity, so to speak. So in the case of translational research is kind of this special niche where we can do research with real patient data, real patients on real patients in real time, and we can do the experiments in the lab, take those results, go back to the patients, and try and figure out, can we help them better? We don't have to go through the clinical protocols that the biotech companies do because we're not technically deriving a drug or trying to bring something to market. So translational clinical research is a fantastic way for you to be a scientist but also have one foot in the clinical door, so to speak. Right? And so in order to be able to do clinical translational research, typically you have to be working with an MD, somebody who can see patients, refer patients, give you clinical phenotypes, things like that. And so Dr. KRUER is, he's the director of the Pediatric Movement Disorders Clinic at Phoenix Children's Hospital. He's a board-certified pediatric neurologist. So most of the patient data we're going to be talking about is data that he's obtained throughout his career. Now, we're also trying to, so cerebral palsy research, genomics research, is kind of in its infancy. It hasn't been happening for very long. Right? So most of what I'm going to tell you today is fairly new, like within the last 4 years data. And so most people, even in the clinical industry, don't know this stuff yet. Right? So you're among the first. So in order to foster kind of international relationships, we started this international consortium. There's only about 4 or 5 labs worldwide that do cerebral palsy genomics research. Right? And we are one of them here at PCH. And so Dr. KRUER is also the chair of the International Cerebral Palsy Genomics Consortium. There's a meeting for this coming up in April. It's in China. Why don't you guys come? Okay. So the actual lab, the research facility, is located in the Arizona Biomedical Collaborative. Has anybody ever been to TGen downtown? So we're in the same parking lot as TGen. So our big building called ABC1 shares the visitor parking lot with TGen. So we're right smack dab downtown. This is a collaboratory science building. We have PCH lab members. We have U of A College of Medicine faculty. And we also have ASU as well, the Nutritional Sciences and Dietetics program. Most of their research labs are located in that building. So there's a lot of stuff going on there. Okay. So before we get to CP, all right, cool. Before we get to CP, we need to talk a little bit about neurobiology. Right? So the last 20 years of neurobiology has been really exciting. It's led to one really important conclusion, that the human brain is really, really, really friggin' complicated. That's like the takeaway message for the last 20 years, right? We've been doing research on the brain for a really long time, and we still have no idea how it works. We know bits and pieces, but the overall concept we don't really grasp, right? This image is a bunch of rat neurons. And the rat neurons are in a petri dish, and they're growing. When you put a bunch of neurons, you take them out of the brain, you throw them in a dish, they self-organize into these little mini brains, I like to call them. And they reach out their little axons and dendrites, and they connect to other little mini brains around them, and they start communicating. Right? So this is about 5 million neurons in a petri dish. Okay? Does anybody have any idea how many neurons the human brain has? Anybody? 84 billion. 84 billion neurons are in your brain, and about an equal number of supporting cells, about 84 billion. So you've got about 164 billion cells in your brain all doing this, and we're trying to figure out how it works, right? And it's amazing to me that so many of us are normal, and we have functioning brains, because the amount of things that could go wrong with 84 billion cells is mind-boggling, no pun intended. Okay? So we have to talk a little bit about CP now as a kind of a clinical definition. What actually is cerebral palsy? The first thing I want to ask, and this is kind of a general survey, how many people know somebody or have somebody in their family with CP? Look around the room. This is a disease that affects a lot of people. And we'll talk about some of the statistics soon. How many people know what causes CP? Does anybody have an idea? What were you taught causes cerebral palsy?
Something that happens during birth in the uterine environment. They had a period of hypoxia.
Same thing? Yeah? Lack of oxygen to the brain, right? So keep that in your mind. That is what almost everybody currently in academia and in the clinical field will tell you causes CP. cord wrapped around the brain, infection in the uterus, child has CP all the time. Okay, so just keep that in your mind. So cerebral palsy is a group of permanent, non-progressive neurodevelopmental disorders, and it's categorized phenotypically by a rather diverse set of phenotypes, right? And when I say phenotypes, it's what is the student or what is the patient look like? How do they act? How does their body move? That's what a clinical phenotype is. Dystonia. So dystonia is the ability to control your muscle tone, right? Dysphagia is the inability to swallow correctly. Torticollis is when the muscles of your spinal tract and abdominal tract, the front don't kind of coordinate correctly, and so you go like this. You might have seen people with CP. They often turn their head without wanting to do so. That's called torticollis. Spasticity, so jerky movements, right? So inability to control all of your motor units and your muscles. Ataxia, the complete inability to walk correctly, right? Poor coordination, so not realizing where you are spatially, which is primarily your cerebellar function. And then paralysis occasionally, or combinations of all of those. Right? There are many associated impairments, and I just mentioned a couple of them. So 1 in 3 people with CP are unable to walk. 1 in 4 unable to talk. 3 in 4 experience pain in some way, shape, or form, like phantom limb pain or pain from their muscles being contracted all the time, different types of pain. 1 in 4 has epilepsy, so seizure syndrome. 1 in 5 has some kind of a behavioral disorder, ADHD, ADD, something like that, OCD, Tourette's, something like that. 1 in 2 has an intellectual impairment. So about half of all CP patients are intellectually disabled in some way, shape, or form. They have a learning disability, something like that. 1 in 10 has a difficulty seeing correctly. So they have some kind of an ocular degeneration or an ocular malformation. 1 in 4 has bladder control issues. 1 in 5 has just some kind of a sleep disorder, insomnia. And 1 in 5 also has some kind of a salivary issue. So all of these comorbidities is what they would be called in the clinical context. They occur because of the region of the brain which has been damaged. Okay. Now, there are many types of CP, and the type of CP is often determined by where we can see the damage occur. We can't always see the damage in CP, and that's kind of what I'm going to focus on in a little bit. So here, spastic CP is typically associated with damage to the parietal lobe. Right? So the parietal lobes of your brain is here. Sometimes it can be temporal, but most often it's parietal in nature. And that can lead to hemiplegia, diplegia, quadriplegia, or disabilities in these particular appendices. We can also have dyskinetic movement CP, which is where they can move, but they can't control how they move. Right? And typically this is some kind of a lesion in the basal ganglia, which is the part of the brain deep within the middle that controls the coordination of all the incoming movement signals from your body and to your brain. Right? And then ataxic cerebral palsy is typically caused by a lesion that's visible in the cerebellum or the tracts leading from the cerebellum to the rest of the body. Okay? All right. So before we talk about why we need to redefine cerebral palsy, we have to talk about kind of the state of CP, the science, what we knew 10 years ago and what we know now. And I want to keep this an interactive discussion. So if you have any questions, just raise your hand and I'll answer them during the whole club. Yeah?
So for instance, how you said that what's been researched on what the causes of cerebral palsy are, is it hereditary?
We're going to get there. Keep that question in your mind. But the answer to that is yes, it can be. So we will talk about that. So let's look at kind of where CP has been and where it's going. Right? So 1 in 250 children born in the world have cerebral palsy. It is the most prevalent neurodevelopmental disorder in the world. Okay? We've known about it for a really long time. We've been doing research on it for over 100 years. We have known, and it's been well established in the community, that lack of oxygen to the brain, intrauterine infection, or fetal damage to the fetus can all lead to CP. Well documented. Okay? But being that as it is, we've had measures in place to counteract these things for about 50 years. So if you look at the overall CP prevalence, the number of new cases of CP, right, or the number of total cases per year, reported per year, and the cesarean percentage. So we do C-sections, emergency C-sections, typically when there's fetal distress, lack of oxygen, hypoxia, damage to the uterine wall or the womb in some way, shape, or form. And the cesarean oftentimes increases the life expectancy and prevents cerebral palsy in the cases where hypoxia is indeed the cause. Okay? But look at this. Wouldn't you expect that if the primary cause of cerebral palsy is hypoxia, an injury to the fetus, that we would see a decrease in CP prevalence? Right? You would expect that. That would make sense. If the primary cause of cerebral palsy was indeed lack of oxygen to the brain or damage to the fetus, we would expect to see a dip here which is inversely proportional to the cesarean percentage. Right? Does that make sense, everybody? Right. So we don't see that, right, at all. Right? And so currently still, even in light of this data, which we've known about for a while, most clinicians still tell patients that CP is caused by damage to the brain during birth and hypoxia, right? And it's just not the case any longer, right? And so what kinds of changes do we need? We need to disseminate this information to the clinical community. And this is one of the reasons why I'm talking today, is because you guys are future clinicians, right? Who runs or who makes sure that clinical establishments run at their peak performance? Is it doctors? No, it's nurses, right? So nurses are the ones that make sure that clinical establishments run as a well-oiled machine. Good nurses is a good practice. Right? Okay, so the current data, and so somebody, so this is one of our collaborators here, Karen, and so one of them said, okay, let's look at all the data, and let's analyze all the data that's currently out there in the literature, and what does that tell us? Right? And they came up when they say, well, none of the data actually points or supports that most of CP is caused by hypoxia, intrauterine hypoxia. And so they're actually saying that most likely the CP is actually due to something that occurs before birth, during development, right? Not some kind of an intrauterine insult or hypoxia at all. Right? And what's kind of the importance to this? Why should you care about this, right? And so this is one of the things where a mother has a child born with CP. She goes and she Googles cerebral palsy, and the definition of cerebral palsy, or the causes, come back from WebMD as malpractice, your doctor did something wrong during birth, or you had hypoxia, right? So if the child had no symptomology of hypoxia, then the mother immediately says, the doctor effed up my birth, right? I'm going to sue them, and I'm going to sue the hospital for malpractice, right? And so billions of dollars in medical malpractice fees have been awarded to CP patients. And so billions of dollars. So this is an inflation in healthcare costs. And as we're going to see here in a minute, really only 10% of all CP cases are caused by asphyxia and hypoxia. Right? So that means 90% of the billions of dollars here was awarded incorrectly. That's a staggering number. Okay, so can we actually determine that mutations in genes cause cerebral palsy, right? Are there other neurodevelopmental diseases that are caused by gene mutations? In fact, there are. And so several of these particular disorders have set the precedent, right? And autism has been in the spotlight a lot lately. There's a foundation that started called Autism Speaks. They are one of the largest nonprofit organizations that have been helping to fund autism research for the last 15 years or so. And there were physicians involved in autism clinical care, and they said, something's going on here. Again, with autism, most people thought it was environmental, or they thought it was vaccines, right? Which there's absolutely no scientific evidence to support vaccines cause autism at all, ever, right? And that was some sensational things by Jenny McCarthy, right? Crazy, crazy loony person. Okay, so now, after we started doing these sequencing analyses, these genome-wide association studies, we're seeing that there are over 100 genes that can lead to, combinations of these genes, mutations in these genes that can lead to autism. Is it always mutations in genes? No. We know that most of these disorders, although there's a significant genetic component to these disorders, there's always some kind of an environmental stress, right? So we are 60% genetics and 40% environment. So genetics can predispose us to a condition, but some environmental characteristic or can push us over that edge. Right? Schizophrenia, we've identified over 100 genes that can lead to the development of schizophrenia. OCD, we've discovered there are certain combinations of genes that can predispose one to obsessive compulsive disorder. And Parkinson's, we have narrowed it down to just 18 genes, and 3 to 5% of these are established as monogenic causes. What does that mean? That means you can have a mutation in one of these 18 genes and you will get Parkinson's. Right? So we know absolutely that there are cases where a single mutation in one gene can lead to the development of a neurodevelopmental disorder. Right? So yeah.
So is that how they like diagnose? Like, do they take those 18 genes that they know cause the Parkinson's?
Yep.
And then they take those genes and they test each of them
Yes.
to figure out, and can they figure out which gene specifically caused your Parkinson's?
Yes. So they have a panel, they take your DNA out if you have Parkinson's, they sequence your DNA, and they figure out which mutation and which gene you have that causes Parkinson's. Right? Not every time is it those 18 genes. So there's other things that are going on too, but we know for sure if you have this mutation in one of those 18 genes, you're going to get Parkinson's.
You said one gene, is it possible that 2 of those 18?
And it's also, well, in this particular instance, if you have a mutation in one, you're going to have Parkinson's. If you have 2 mutations, you're probably dead. Yeah. So there are many genes. If you have a mutation in a gene, you're not viable embryo and you spontaneous abortion. So most people who spontaneously abort a fetus, it's due to a mutation that is not tolerated, right, in the biochemistry of the cells. So if you have 2 mutations, you're probably not going to survive. That's the way it works. So as it turns out in CP, another one of our collaborators, Alistair, he's in Australia, he did this huge study, and he says, looking at the actual genetic data, it looks like that the causes of almost all CP originate prior to birth and have nothing to do with malpractice during the birth process. Right? Greater than 30% he found, and maybe even as high as 40% of all cerebral palsy cases is caused by an identifiable genetic mutation. Right? That is a complete change in the paradigm of how do we treat CP, how do we diagnose CP, and why is CP such an umbrella term, right? There are so many different types of cerebral palsy. It doesn't make sense there would only be one cause, right? One cause, if it's a single cause, and everybody who has CP has that one cause, all of their symptoms should be similar. Right? That just makes sense. But we don't see that. So there are probably multiple genes that cause cerebral palsy. And in fact, that's what we're seeing, and that's what we're going to look at here in a minute. So only 10% or less than 10% of all CP cases are due to delivery error. Wow, right? So that's 90% of those billions of dollars that have been awarded usually in cases where it had nothing to do with the poor doctor doing delivery. It has to do with genetics. So only recently has this data started to begin to change the current definitions of cerebral palsy. So this is verbatim from the Cerebral Palsy Alliance website. So they're like Autism Speaks. They're kind of helping fund cerebral palsy awareness and research and things like that. And they still put in there that CP is caused by brain damage, but they kind of allude to the fact that it happens mostly in the womb, not through negligence, and that less than 10% is caused by malpractice or hypoxia. Right? They still don't write in here CP is genetic. So they're not fully supporting the current research paradigms. They're just saying, look, we've got some data, and maybe you should pay attention to this, doctors and nurses in the clinical field, that CP is not caused by the things that you were taught and that you think CP is caused by. Okay, so any questions on cerebral palsy as a disorder or kind of what we're seeing before we actually get into some of the science? All right, yes?
So, for instance, in Parkinson's, even though you've got the mutation, you're born with it, why the delay in expression?
So many of the genes are maintenance genes, and they are maintaining axonal integrity, and so it takes a long time for the degradation to manifest.
Whereas with cerebral palsy, it's manifest.
It manifests as soon as development occurs. It's the wiring isn't occurring correctly. And so there's really nothing you can do about it once birth occurs. Right? Yes?
Now, is it possible that somebody that, let's say their child has cerebral palsy, but they don't show any, the parent doesn't show anything, would you be able to take a look at the parent's genomes before they get pregnant, kind of like Tay-Sachs?
Absolutely. And that's one of the things we're working on, and we're going to talk about that specifically. So that's a great question. So what do we want to know? What is our group focusing on? What did Dr. KRUER have that aha moment, which we'll talk about that moment in a minute. But we want to know, right, what is causing CP that otherwise in a clinical setting has no causes. So what if you have a child with cerebral palsy, but there's no MRI evidence for a brain malformation, and you had a normal birth, non-cesarean, non-premature, no evidence of hypoxic ischemia during your development period? That's what we're interested in. We want to know, of all that non-10% cases of CP, what's causing all this CP? And that's kind of what we're looking for. So we are including affected individuals. We call, in these kinds of studies, genomic studies, we call them probands. So usually we de-identify them, we give them a number, so that way we can share information without sharing PHI, right? Because that's confidential, right? So we include infected individuals, and we want uninfected family members, right? This is important because this is the way we can tease apart where did the mutation come from. Is it inherited from a parent, or is it something that happens spontaneously during development of the fetus? We usually look for probands that have no abnormal medical history, so patients which don't show an MRI finding, or they weren't a cesarean or premature, right? So are there forms of CP that are directly related to patient mutations in this particular patient cohort? So we're kind of what we want to know. Are these mutations spontaneous or inherited, right? Now, because of the intrinsic mutation rate in our own ability, so eukaryotes, to replicate DNA, every single human being has between 300 and 500 mutations in their genes, right? So how do we tease apart the mutations that you're supposed to have? This is an evolutionary driver, right? This is what drives evolution, is DNA mutation. How do we tease apart the mutations you're supposed to have from the mutations that cause a disease, right? And we'll talk about how we do that in a second. So if we do find these mutations, are different types of cerebral palsy associated with different mutations, right? So if you have spastic ataxic cerebral palsy, do you have one set of mutations? If you have completely ataxic cerebellar CP, do you have another set of mutations? We're trying to categorize all these different types of CP by saying, look, we know the genes that your child has mutated. That's why they have spastic cerebral palsy. How are the discovered mutations actually leading to CP? This is the most difficult question to answer, right? So once we identify a gene that shows up in people who have CP, right, what exactly is it about that gene that influences the malformation of the brain? There are a myriad different things here, and it takes some really super sleuthing molecular biology to kind of figure this out. And we'll talk about a few examples of how we can actually figure this out in a second. Okay. So what was the aha moment? What made Dr. KRUER, because he's the pioneer in this field, what made Dr. KRUER say, gosh, there's got to be genetic component to CP? So he was seeing a family from Jordan, the country, a Jordanian family, and the parents are consanguineous, right? That means they are related by blood, right? This happens a lot in nomadic tribal people of the Middle East, especially where they try and keep their family unit in one tribal setting, right? So this is a great place to study human genetics because the genetic background is normalized. We don't have any introduction of new genes into the genetic pool, right? This is also a bad thing for the longevity of families, right? So interbreeding and breeding with people who are blood related is typically not a good thing for the fitness of your progeny, right? And this is a perfect example of that. Two normal, otherwise normal parents, they have 5 kids, 4 of them have CP, right? So that's a red flag right there. The chances of you having 4 children that have a mutation or that have a hypoxic event that causes CP is like 0, right? Pretty close to it. But the chances of this being heritable is very high. And that's exactly what the pedigree looks like, right? And if you notice, females and males, there's an equal ratio here. 2 males, 2 females. So at first glance, it doesn't appear to be sex-related. Okay? There is a slightly higher prevalence of men who have CP than females, right? So there is a little bit of an X-linked there. So in this case, he started doing some of these genetic analyses with this family. He sequenced the parents, and he sequenced all the kids, including the unaffected individual, and he found that all of the affected individuals had a homozygous mutation in one gene only. Right? So what does homozygous mean? Same. So in DNA, how many strands of DNA do we have? 2, right? And how many copies of each gene do we have? 2, one from mom, one from dad. Okay? Homozygous means the mutation was in both copies. Right? So the copy that you got from mom and the copy they got from dad both had the same mutation in the same gene. Right? So that indicates that the genes, one copy from mom, one copy from dad, were inherited to the kids. So any child who got both bad copies had CP. Right? So this was the very first indication in 2013 that CP, or the not indication, but the first shred of good evidence that suggested that CP was indeed genetic. And it turns out this particular gene messes with the cytoskeleton. This is the network of proteins that keep the cell, kind of gives it its shape, and it also controls how neurons migrate and connect to one another. Right? So we mess that up, we can mess up the brain, basically. Right? So this was the first real evidence that, hey, yeah, we can cause CP by disrupting one gene, and one gene only. So we can see I'm going to turn the lights off for just a minute here because, holy crap, okay. But we can actually see this much better, all right? So these are MRIs. Now with CP, there are instances where you see nothing. You can have a patient that has severe cerebral palsy, and you see nothing in the MRI. The MRI looks exactly like a normal person. Right? But occasionally, we can see that in an MRI, we can see that, for example, these are hyperintensities here. So these areas that are brighter white in this particular type of MRI, they indicate a hypoxic ischemic event. Right? So in this particular case, we have a hypoxic ischemic event which occurred, which led to this patient's cerebral palsy. So these types of patients we would exclude from our study. Okay? Now, this is a little bit different. This is severe brain abnormality. So these are the 4 children from that Jordanian study. These are their MRIs. And you can see severe deformities in the corpus callosum of 2 individuals. You can see some hyperintensities here next to the basal ganglia. And you can see that this particular child has a much smaller brain than normal, and it has way more white matter than gray matter, as well as ventricular megaly. So these are the lateral ventricles inside the brain. They're supposed to be on the smaller side, so kind of like this here, and they're much larger here. So these are gross abnormalities caused by the genetic mutation in this instance. Okay? So how can we show this? How can we actually do the science? How can we figure out and tell you for sure that this gene that we discovered causes cerebral palsy? There's a process here. There's something that we have to do scientifically to show this with certainty. Right? So what do we have to do? Well, first of all, we have to sequence the DNA. Right? So we do this. These are called trio studies. So in order to figure out what happened, where this mutation came from, we have to look at the parents, right? Because all the DNA comes from your parents. So we look at the mom, we look at the dad, and we look at the proband, right? And if we can, we get an unaffected sibling. And we use that as a control, right? So we sequence the DNA, and then we use computers, bioinformatics, right, to identify rare alleles, right? Because if we see, so we have databases, we sequenced hundreds of thousands of humans, and we've deposited all their genetic information into a database. It's all de-identified. We don't know where they come from. We don't know what they are. We know race, age, and sex. That's about it, right? And so we can search those genetic databases, and we can look at all the different mutations that occur in a population of humans. And we can say, okay, well, 40% of the human population has this mutation. Do you think that mutation is going to lead to CP? If 40% of all humans have it, do you think it's likely that that mutation is going to cause CP? No. It's super common. That would mean we would have way more cerebral palsy. So we only want to look at the mutations that are identified that have a very, very low frequency. We're only interested in the super rare mutations that occur because, after all, that's what we're looking for in CP. Right? OK, so then once we identify a mutation or more than one mutation in these patients, OK, we have to say, well, all right, so we identified a mutation in the computer. But that's not good enough. We have to show that if we use this mutated gene, or the protein that's produced by this mutated gene, do we see something go wrong inside some model system? Okay? Now, if I had a flyer up and I was looking for potential people to participate in my study, and I'm going to mutate your genes, are you going to sign up? Absolutely not. So we cannot do this in humans. We can't take a normal human, mutate their DNA, and see if they get CP. Okay? In 1945, maybe. All right? But nowadays, we can't do that. All right? So we have to use other model systems to kind of model this human mutation and see if it does mess something up at the molecular level. Right? So this is one of the first ever mutations that were validated that if you have the mutation, you get the disease, right? And there's a homozygous versus a heterozygous version of this, right? Whether you have 1 copy or 2 copies. And in 181, 182, and 201, we talk about this, right? This is sickle cell anemia, right? And so it's a single base change in DNA. Right? We can see that here. This is wild type. This is what it's supposed to be. And if the mutation occurs right here, right? It changes the mRNA, which changes the protein. We have the sickle cell version of hemoglobin. Right? So we validated that at the molecular level. We've identified that by DNA sequencing. We know that if you have this mutation, you're going to have a version of sickle cell anemia. Right? So this is one of the oldest. This is just an example of kind of what we're hoping to do with genes involved in cerebral palsy. Right? So how do we do this? What's the process? And this is the same process for autism, OCD, anytime that we want to figure out whether or not a mutation causes a human disease. This is the basic process that we're going to go through. Right? So we get our DNA, and we're going to do something called whole exome sequencing, WES. Right? So how much of our DNA is actually made into a protein? Does anybody know? Take a guess. What percentage do you think? So we have 3 billion A's, T's, C's, and G's inside of every one of our cells. What percentage of that 3 billion do you think actually makes the proteins which you can see as us?
25.
25? Who else? Anybody else? 1%. 1%. Only 1% of our DNA actually codes for a protein, right? Very small number. And we are interested in mutations in only the parts of the gene that make proteins at this point. Now, me as a scientist, I'm really interested in what happens in the other 99% of the DNA, but that is much harder to tease apart. Right? So right now we're going for the low-hanging fruit, and we're focusing only on the protein coding portions. And that's what whole exome sequence does. We take a person's DNA, we shear it, we break it up into millions of fragments, and then we pull out the fragments only for the pieces that code for protein. That's called the exome. Right? Exons are the pieces of a gene which make a functional protein. So we sequence all of the protein coding portions of the individual's genome in this machine here, and then it gives us out this huge file with a bunch of A's, T's, C's, and G's. There's not really much you can do with this. So we need some kind of a computer specialist, right? So they take all this DNA, and this is called a bioinformatics expert. We'll just say that, right? There's another word for it, bioinformatician. Yeah, bioinformatician. Right. The word, well, I lost it. So you have the trio DNA. You got your sequencing. We pass it through a bunch of these computational filters, and we compare it to the databases, right? And then we say, OK, which of the mutations that we detected in these probands, which ones are the rarest? right? Let's focus on the rare ones because those most likely are the ones that are leading to these relatively rare clinical phenotypes, right? And so we do a whole bunch of filtering, yada, yada, and we figure it out. Is it de novo, of the new in Latin, right? So that means that it's a mutation that occurred spontaneously. It wasn't passed on from parents to children. That happens quite a bit. So there's, like I said, every single person in here has between 300 and 500 de novo mutations, right? And so maybe one of those 300 to 500 happen to fall in a gene that you can't mutate, right? So that is the case and sometimes it's a biallelic, right? So the parents both have a bad copy, the kid gets 2 bad copies of the same gene, and boom, they have a clinical phenotype. Right? So we can do this computer analysis and we can figure that out. Right? So this is just an example. So trio 1, trio 2, trio 3, so on and so forth. We collect all these trios, we analyze them, and we get a list that pass our filters of all these potential genes that can cause a problem. Right? And then we compare all these variants and we look for commonalities. Do we see the same gene in multiple patients? Do we see the same biochemical pathway? In other words, are there a bunch of genes that control the cytoskeleton? Do we see those mutated more often? So what's the commonality? What's the common thread here? And that's going to help us later. So if we look at an example, this would be kind of the output of DNA. Right? This would be the output of a single trio. So mom, dad, dad, mom, son, affected individual. And then these are a list of the potential mutations that pass our filters. Right? And so we get a gene. Here's the gene name. This is a relatively unknown gene. We don't really know what the function is. But this one we kind of do know, and this one we kind of don't know. But these are the frequencies. So this is pretty common gene. These are not so common, and this is pretty common. Right? They pass our filters, but they're on the more common side of our filters. So we have to then, as scientists, use logic and say, okay, most likely, it's going to be this guy. Right? And if you notice, this is a mutation that's inherited from both parents. Right? This is a compound heterozygous mutation. That means there's a different mutation in each copy of the gene they inherited. Right? So this is a pretty bad mutation, and this leads to cerebral palsy. Okay? So if we look at all of our data, 164 trios we've got data for. So 164 times 3 plus a couple of unaffected siblings that we've got there. And so we've got 86% of all the affected individuals have greater than 1 predicted mutation. 109 identified were de novo, pretty significant, right? So here, these are X-linked. So this is where I was telling you that it seems like slightly more men get it than women. Why is that? There's a mutation on the X chromosome. Why does it affect men more than women? Men only have 1 X chromosome, right? Women have 2. So you can get a bad copy from one of your parents, but you get a good copy from the other parent, and then you don't get it if you're a female, right? So men got the short end of the stick on that one, unfortunately. So on average, we identify that there are about 2 to 2.5 mutations predicted deleterious. So that means that the mutation that they have that we looked at is bad, right? Got it? Sorry, somebody dropped something. And so that's pretty significant, right? We've shown computationally that almost all of the CP patients that we're looking at have a bad mutation, at least 2 bad mutations, right? So now how do we go from that? And we're running short on time, so I'm not going to talk about this, but you can ask me questions later. So how do we go from that to showing that these genes do, in fact, cause a problem? Right? And we can use lots of different models. All right? And it's preferable when you do these molecular analyses that you use as many models as you can. Right? So we can use flies, Drosophila. We can use fish, Danio. We can use yeast, Saccharomyces. We can take cells from the patients themselves, and we can grow those cells in petri dishes, and we can do in vitro analyses on those cells, things like that. Okay? So I'm going to just go through 3 examples here, just quickly. Row B, PNPLA7, which is the gene we just saw, and MKL1. These are 3 genes that we know kind of what the function is in humans, but not really that well. So we don't really know why they would cause CP, and that's kind of what we want to figure out. Right? So the first couple of things I'm going to look at is yeast that I'm going to show you. So yeast is very easy. It's super simple to grow. Grows on petri dishes. It will grow no matter what for the most part, and we can manipulate genes in yeast. Okay? And yeast are very, well, I mean, a couple of billion years related to us. And so they actually have some of the same genes that do the same things that we have. And so if the gene that we identify has what we call an ortholog, a gene in Saccharomyces that is evolutionarily related to the human gene, we can manipulate that gene and see what it does to the yeast. And sometimes we can take the human gene and put it into yeast and see what happens. Right? So that's kind of one thing we did. So these are colonies of yeast that we have genetically manipulated. And so here we've taken the mutation that we discovered in humans and we mutated the gene in yeast, the same gene, with the same mutation in the same spot of the protein. And it doesn't grow. Right? So that's showing that if we take this mutation that we identified in the computer and we mutate the yeast protein and we try and grow it, it doesn't grow. And then here we can actually put the human mutant protein in. So we removed the yeast protein and we put the human version of the gene in and the mutation, right? And it grows. Right? So that means that this protein right here is not functional because it's not killing the yeast, right? In this particular case. Right? So one, we're showing that if we mutate the only copy of the yeast protein, the same mutation, the yeast dies. And if we put in the human version, right, of the protein with the mutation, and we leave the yeast protein in there, it lives. Right? So that means that the human protein is non-functional. Right? So we know that it's messing up the function. Right? We can also use flies. So we can do similar studies in flies. Right? And so flies actually have a very similar pathway of brain development that humans have. And we can watch individual neurons in a fly. We can watch how they migrate, how they develop, and how they make connections. We can also look at locomotor phenotypes in flies. So if we mutate a gene, does it affect how the fly flies and walks? Right? And so indeed we can. So here is an example. So on the left here, we've got our control flies. These are normal flies. And on the right, we mutated or deleted the gene in flies called MRTF. Right? And so when we do that, what we're going to measure here is we're going to measure the speed at which they can crawl. So we knock them all down. Now watch. Crazy, right? These flies have CP. They can't walk up the tube. And so we can quantify that. Here we go. So the flies without the mutation, they go really fast. The flies that have the mutation are significantly impaired. Right? So that's a big nail in the coffin for this gene. Look, we mess up this gene in flies. They can't walk. Right? And that parallels what we see in humans. Right? We can also look at human cell lines. So we can take the patient lines and we can convert them into something we call lymphoblast. And we can look at proteins and shapes and the way they grow and see if there's differences compared to a wild type. Right? We can extract the mutated proteins from the patient cells and we can compare enzymatic activity. Does the protein actually work? Does it bind to what it's supposed to bind to? Things like that. And we can look at the gene and when it's expressed in certain cells. For example, ROB, we really didn't know what it did in neurons. But here I did an experiment where I looked at the expression of the ROB gene in human neuron cells. And so I started them off as stem cells, and then I coaxed them to grow into functioning neurons in the petri dish. And ROB goes way up. So when they're a stem cell, there's no ROB turned on. But as soon as you start differentiating them into neurons, which is what happens during fetal development, the ROB gene goes up. So that's telling us that ROB definitely has some major function in neural development, which we didn't know before. Right? So we can also look inside of cells and look at the proteins and what is the mutated protein doing to the cell. Does it change the shape? Does it alter anything about the cell? So here we have wild type ROB. Right? And these are COS7 cells, which are monkey cells, and we grow them in a petri dish. Right? And so we can put the protein inside, and we can look at it. So here's ROB and where it's located. Here's the mutant ROB. Looks like there's a lot of it, a lot more of it. Right? And it's mostly in the center. And then there is some evidence in the literature that suggests that Rho-B actually regulates actin in the cytoskeletal network. Right? So it's important for how cells move and how cells are arranged and what their shape is. And so if you look here, you can see there's a big difference between the actin in these cells and the actin in these cells. Right? It looks highly dysregulated. Right? So that's another kind of, that's a nail in the Rho-B coffin saying, look, we put in the mutated version of the human protein in monkey cells, which they have 98.5% the same DNA we do. We put this mutated gene in monkey cells. It messes up the actin. It's something we can see and we can visualize. Okay? And then I can also say, OK, I'm going to take human cells. So these are human fibroblasts. And I'm going to turn off the Rho-B gene. So what happens if I turn off the normal Rho-B gene? Normal cells, normal actin. So actin's in green. Look what happens. They get all crazy. Right? They get long and thread-like. They almost look like neurons, kind of. Right? And so that gives us an indication that Rho-B is indeed controlling how the cell looks and moves. Right? PNPLA7, so we have 2 patients, and they each have mutations in 2 different parts of the gene. Right? So these are domains, and we're not going to really get into what those are. We have 2 patients, 2 different spots of the protein. So this is the protein here. And so what we can do is we can take their fibroblasts, and I can turn them into fat cells. So I looked in the literature, and I said, well, PNPLA7 has been shown to be involved in fat cell development. So if we look at these patient cells and we try and make them into adipocytes, fat cells, do they look different? Is there some kind of an issue? Right? And this is normal, right? Normal human cells. Right? These are lipid droplets inside of cells. Right? And the more red the color, the more lipid is inside. Right? So this is one patient. Does it look any different? Not really. We don't see much of a difference here. Does this one look different? Much different. So the lipid droplets never quite get as big. The lipid droplets also never get as full of lipid as these guys do. So at least one patient in this particular assay shows an aberrant phenotype, right? Showing that if you mutate PNPLA7, PNPLA7, you get some kind of a disorder. Right? OK, great. How does adipogenesis, making fat cells, how does that have anything to do with cerebral palsy? How does messing up the cytoskeleton have anything to do with cerebral palsy? That's the piece we're missing right now. How do we link what we found at the molecular level to CP? And that's, after all, what people care about the most, right? Great. You found my kid's genes mess up fat development. Well, my kid's skinny. Right? How does that have anything to do with cerebral palsy? Right? And so that's kind of what we really are focusing on now. And so what we can do is we can take stem cells that we take from the patient. So I can take your cells, and I can do this to anybody. I can take your cells, and I can turn them into stem cells in the lab. Then I can take those stem cells, and I can turn them into any cell type in the body, including neuronal cells. So I can mutate DNA in those cells, or I can correct the mutation that's in those cells and see how the neurons develop. Okay? So this is an example of where I took some wild-type cells, some normal human stem cells, and I turned them into neurons. And this is kind of a picture of that neuron. I've taken out all the background, and I've shown you only the dendrites and the axons of that neuron. The asterisk is where the nucleus would be. So this would be the soma of the neuron. Right? Remember, you're 201. Right? So the soma of the neuron. So I'm going to tell you right now, everything you learn in school, if you're in the sciences and the clinical industry, you're going to use. I promise you. Knowing microbiology and knowing your A&P is paramount to your career. And so we've got the nucleus here in the soma. But this, if we take the same cells and we put the mutant version of Rho-B into those neurons, look what happens. Right? The neurons don't know where to go. They don't make long axons. They don't make long dendrites. They don't what we call arborize. They don't make all these little stems that come off. Right? They're all messed up. Right? And so right there, that's our link for Rho-B. Look, we've demonstrated in flies. We've demonstrated in yeast. We've demonstrated in cell culture, both in monkey cells and in human cells. And now we tried to make neurons with this mutant, and we can't. They're bad. They don't work right. There's our cause for CP. If you have a Rho-B mutation and it causes a non-functional protein, your neurons can't attach to other neurons correctly, and that leads to the developmental disorder we see. Right. All right, so in summary, so let's just talk about what we did. So whole exome sequencing is a great tool. It pinpoints contributing mutations in when we're doing genetic studies. But just because the computer says it's a mutation doesn't necessarily mean it is. And so we have to what we call molecularly validate that mutation through all the different things that I just showed you. Okay? So CP is primarily a genetic disorder, which is a complete 180, right? So CP is mostly caused by mutations now. There's about 14 to 15% of CP that we can't account for a cause for yet. We're working on it, but most likely that's in the other 99% of the DNA that we can't look at yet. Right? And so usually it's a spontaneous mutation, 35% of the time, or it's typically inherited by an X-linked pattern. So 36% of all CP is inherited as a faulty X chromosome or a gene. Right? So our functional validation assays, they have shown that there is a significant impairment at the molecular physiological level for Rho-B, PIN+7, and MKL1. So we have now established that mutations in these three genes are definitive causes for cerebral palsy. Right? And then identification of genes definitively involved in the pathoetiology of CP will help us lead to targeted in vivo therapies. Right? So again, a lot of you might be asking, okay, well, that's great. You figured out what caused my kid's CP, but what can you do about it? Can you do anything about it? Is there something we can do to prevent CP? And a lot of you have probably heard of CRISPR. It's in the news. It's the most highly anticipated genetic manipulation tool there is. And so in theory, like for ADD3, that gene that we say causes monogenic CP, if we can identify that you carry that particular gene, we can do IVF, we can take your eggs out, or the sperm, and we can replace that bad gene. Right? We can put a good copy of that gene in and do an IVF, and now you can have a normal child. Right? Or, this is a little bit in the future, people are working on these therapies, but intrauterine genetic manipulation. So if we discover that you're forming zygote or morula, if we do some early genetic screens, has this mutation, we can inject CRISPR-Cas9 technology into the egg, which can reprogram all of the cells and remove that bad mutation. Right? In theory, we can do that. We don't have approval for that now. We can't do the clinical testing for that yet in humans, but they've done things in mice that are showing promise. Right? So that's the potential for how do we treat, how do we prevent cerebral palsy. Okay? This is our lab. I actually got airborne in this picture. This is Michael, and then a lot of the data that I showed you was from various individuals in the lab. This is Samaya. She's our bioinformatics specialist. She does a lot of the computational work. And then we've got our fly specialist, Sarah. We've got a molecular biologist, Aurelian from France. And then Sergio is our yeast guy. He's kind of in the back. And then I kind of do a little bit of everything, and I oversee various projects. So if you have any questions, please let me know. I'm around.